The second part of the evolutionary mechanisms module 4, is looking at sources of variation. And this is really, the explanations, the how and the why, observe to explain changes in gene or allele frequencies over time. So, we're going to cover things like mutation, genetic drift, and two special cases of genetic drift as well, known as the founder effect and population bottleneck. We'll also cover gene flow, and natural selection is another evolutionary mechanism, but we'll spend a whole nother lecture 4.3, on natural selection exclusively. So what is an evolutionary mechanism? Well, it's just a factor that alters variation and patterns that we observe in population samples and over time. This is something, right, that we're studying through generations, because that's part of our definition of evolution: change in gene or allele frequency over time that we can observe. So evolution is a law, but it's, there's also a theory that surrounds it. So evolution is a law, can't prevent it from happening. You're going to have change in gene or allele frequencies over time, and those genes affect phenotypes, certain traits that we can observe and we can see. So we can record, the generation changed, just like in our first example with the ABO blood group. Generation 1, here's the allele frequencies of A, B, and O. Here's generation 2. The population has evolved. Now, to answer how the population evolved, or why did the population evolve, we use these evolutionary mechanisms: mutation, genetic drift, gene flow, and natural selection. What's really neat is we never really have a model that is for natural selection. What we basically do is we reject all the other ideas, and then oftentimes natural selection is the only one that cannot be rejected. And we know from our ideas about evolutionary theory and science that if it cannot be rejected, then we know it's true and correct by default. Let's talk about mutation. And I'm not talking about mutants and X-Men and stuff like this. A mutation is basically just an alteration to the genetic code. It can be one single change. It's oftentimes just a mistaken copying that may or may not lead to a change in an amino acid. And of course, proteins are made of amino acids. Right? Amino acids are the building blocks of proteins. So there can be one slight change, and it may change the protein, it may not. And then if it changes the protein to a certain extent to then affect bodily functions, that's different. But if you look at a mutation, an error in copying or a mistake, we interpret it as either neutral, positive, or negative. We assume that most mutations, for the most part, mutations that occur in organisms are neutral or mostly neutral. That is a number of different traits that don't necessarily affect the organism. You could probably think of a number of different traits that we have that are neutral, like eye color and hair color. There's really nothing that gives us an advantage in the environment. Now, there are things like sexual selection where females might prefer a specific phenotype like eye color, hair color, and you may see those levels fluctuate of that phenotype over time. But that's slightly different. So we talk about neutrality, meaning a trait that we observe in an organism and it may persist, but it doesn't really affect the organism at all. Something that's positive, right, is a mutation or error that occurs that gives a benefit to the organism. There's also there can be negative mutations, a mutation or mistaken copying that occurs that actually is a detriment to that organism. So in the case of this picture here, it's a salamander, and it actually has 3 arms. You can see another arm close to the arm on its right body, kind of a little bit more on top of its head, closer closer to it. So the question is this third arm, right? That's the mutation. Is it positive, negative, or neutral? Well, we can try to think about a number of different scenarios. One, this looks like an animal that's in captive care right now. It looks like it's in somebody's house. So thinking about the environment, an environment with as a pet, or with humans caring for it, or in the wild, right? Let's say in the wild. Let's think about in the wild. In the wild, is this neutral, positive, or negative? Well, one good question is this third arm for the salamander, is it mobile? Can it move around? Can it grasp things? So that's something we have to kind of think about. This is probably either neutral or negative. I'm assuming that this arm is not very mobile. It doesn't work. It may not affect the organism at all. It may not be attractive to females if this is a male, right? They may say, "Oh, that's not something that I want to see in my offspring." That will likely, put them in the best situation to have offspring and perpetuate our lineage. It could also be negative, where this salamander is trying to scurry and move around very small and tight enclosed spaces, and this third arm could get in the way. Right, and then that could be the difference between being able to find food, starving, and possibly getting eaten by a predator or any other environmental factors. So mutation is an error in copying. It may or may not affect the protein or the trait that's being displayed, and then we evaluate that mutation is as either neutral, positive, or negative. One thing to remember is that one mutation in one environment might be positive, and in another environment it might be negative, and vice versa. It could be neutral in one, positive in another. Just think about that. Also, a mutation could be positive in one generation, and it could be neutral, negative, or something else in the next generation. So just think about that. Let's go over some of the blood polymorphisms, specifically in the example of a point mutation. So a point mutation is typically known as a single nucleotide polymorphism, where it's one single point on a gene, right? Nucleotide, which of course we now know is a sugar, a phosphate, and a nucleic base, adenine, thymine, cytosine, guanine, at least in DNA. And we know that thymine doesn't code for any amino acids and thus any proteins. But if you look here, and this happens in humans, there is variation in hemoglobin, and you can have mutant hemoglobin, which is shaped in a sickle, and the sickle hemoglobin has trouble transporting oxygen to the rest of your body, whereas the normal hemoglobin has no problem with this. And if you have sickle cell, then you can potentially have some major issues with iron deficiency and iron deficiency anemia. This follows Mendelian mechanisms of inheritance, where you have one point on a gene responsible for a physical trait, and you have to have two traits, right? Two alleles to express the trait. So if you have normal hemoglobin, you have both copies of normal. If you have sickle cell traits, you have one normal copy and one sickle cell copy. If you have sickle cell disease, you actually have, you're homozygous recessive, you have both copies on both your chromosomes and those genes of the sickle cell allele. You can notice here that in DNA, the code for normal hemoglobin in this specific part of the genome is CTT. But one single change in the middle from a T to an A will change the messenger RNA strain, as well as, how you're going to present the codon, the anticodon, right? The three triplet of bases that swing around and stitch the amino acids together to make a protein. So normal hemoglobin would make glutamate here, but sickle cell hemoglobin makes valine. And one single point change can be the difference whether you either have sickle cell trait or sickle cell disease. Sickle cell disease is a major detriment because you have major health problems and there's lots of childhood morbidity and mortality. Plenty of children that die at a young age, particularly in other developing countries where they might not have the capacity that you see in some places for modern medicine. But somebody who has sickle cell trait, matter of fact, has the ability to confer immunity to malaria. And so being heterozygous is actually really good and important in this way. And if you had both copies of the normal hemoglobin, you're highly susceptible to malaria in some of these areas. And being heterozygous is probably the most advantageous. We'll talk about it in greater detail as we move along in the course. I've heard a number of evolutionary biologists that will suggest that not natural selection, but genetic drift is the most powerful evolutionary force. And genetic drift is just random change over time. Yes, you think about it. Evolution is a change in gene or allele frequencies over time. And all we're doing is looking at patterns that are going to change, that inevitable it is going to change. Now, can the change in those gene or allele frequencies be random, meaning that there's really no explanation for a mechanism, evolution, it's just random change? We found no real reason why it's changing other than it's random change over time. Yes, this does happen. Random fluctuations where some alleles can be gained, some can be lost, and these are just random events. Some good examples of random events, even in the environment that cause these changes, are like a natural disaster, right? On an island where a number of different, like a hurricane that comes through maybe a Caribbean island and wipes out a specific species of animal or a particular species of bird, or a bird that had, some specific traits. And what you'll notice is that larger populations are usually less susceptible to the fluctuations of random alleles because there's really large numbers. And if one is lost, that means it doesn't really make a difference. If one is gained, right? These are some of these factors that aren't going to affect large populations. Small populations, it can. Where, if you look at humans, for instance, we have small litter sizes. I have a friend where he is the youngest of 4 boys, right? If you think about it, the chances typically the chances that we would, calculate probabilistically, if you have 4 children, it's a 50-50 chance. And so that would mean we should see an even distribution. That friend of mine should have a brother and 2 sisters, right? And that family having nice, even 2 boys, 2 girls. But why do you have this big swing in all boys? Well, you have small population sizes. It's just like flipping a coin. If you flipped a coin 9 times and you got heads every time, the probability of you flipping heads or tails on the 10th time is the same. It's 50-50 every time. But why it's so skewed to heads 9 times out of 10 right now, or 9 out of 9, is because you have a small sample size. You flip that coin 100 times, 1,000 times, 100,000 times, 1,000,000 times, you're going to be really close to 50-50 because now you have a larger sample size that will get really close to those levels. So that kind of explains small sample sizes always usually look skewed. Large sample sizes are more representative. Let's talk about some of the special cases of genetic drift. The first one being the founder effect. The founder effect is a really neat phenomenon. This is where a small group or even one individual becomes a founder of a population being one individual, in this case, in many other cases, that, or a small population, a group of individuals in one specific, group of people that actually find or are the founders or contribute a lot of information genetically and phenotypically to the next generation, and it's invariably changed. So an example I usually like to give is, the Mutiny on the Bounty. It's actually an old black and white film you might catch on AMC once in a while. And it was about an island and a mutiny between, a number of islands like the Pitcairn Islands and Tahiti. And, these are islands that are in parts of the South Pacific. And you had a number of different individuals from European countries that were part of a number of different trades out in the South Pacific. Matter of fact, I think they were trying to harvest sea slugs in the sea slug trade because they're a delicacy. And this is around the 18th century. And what you notice is that there was a big mutiny. This is where people on a ship have no faith in their captain, and they kind of turn on that individual. And so this happened, and it marooned a number of people on Pitcairn Islands, and they were battling and fighting. And after this battle, only Alexander Smith, who was one of the sailors, a European male, all the women and children on that island. And so here's a picture, in the lower right here, there's a picture of Alexander Smith in the middle. Think about it. And he sired offspring with many of the females. And so any male in the next generation is going to have his Y chromosome, right? All of them, right? So you can see one individual or a small group of individuals can, over even one generation in small population sizes, create a major shift in genotypes and phenotypes. Matter of fact, in some, South Pacific Islanders, we see a degenerative eye disorder that is very rare in populations, in European populations. Well, it was brought by people from Europe, and then all of a sudden we see a preponderance of it in some of these island populations. Population bottleneck is another special type or form of genetic drift. This is where you have oftentimes a natural disaster. It could be earthquake, hurricane, thunderstorm, tropical storm, anything that is kind of a random event that will wipe out a number of individuals that carry a genotype that will not be passed down potentially to generations. And it also changes, right? Changes the composition and frequency of those genes or alleles. And it's because of a random event like weather or climate. The analogy oftentimes is that you have a bottle, and then in that bottle you have a number of different diverse genes or genotypes, and then you pour a little of them out and then throw away the bottle, and whatever you poured out is allowed to kind of produce offspring. That represents the natural disaster, right? You pour a little bit out, those are survivors. The bottle represents those who perished in the natural disaster and will not breed in future and contribute their genes to future generations. You can also see in this diagram that it looks like two bottlenecks touching each other. And this is kind of another example to imagine it. And the example I give here is with elephant seals. And you can see that humans can affect organisms. That's typically seen as a random event to them, not necessarily to us. But elephant seals were almost hunted into extinction. What happened into extinction? Once an animal is targeted, particularly by humans, and you have lower population levels, think about it. Genetic drift also will take place because you have really low population levels. Many of them will become homozygous could be homozygous recessive, homozygous dominant in certain traits, right? And then what happens is you might have mutation blocks that build up, and you could also have a number of different mutations that occur because we know that due to inbreeding, right? So when populations get really small in any organism, evolutionary biologists get very nervous because you have less genetic diversity. Think about it. If you have more genetic diversity, then the organism is best able to adapt, or better able to adapt. Because remember, organisms don't just all of a sudden evolve, right? They don't just all of a sudden, hey, it'd be great if we had another arm, like a third arm, like the salamander, and all of a sudden it happens. No. Organisms have all the genes and suite of characteristics they need to handle things in the future. If you have less genetic diversity, you have a poorer ability to adapt. And this is exactly what happens in population bottlenecks. Gene flow or migration, these two terms are synonymous, is the movement of genes in and out of populations. So we can explain changes in allele or gene frequencies sometimes because of migration. Populations may migrate to or migrate from, right, specific populations. I can give you an example here. The country of Germany actually took the greatest number of Syrian refugees during the Civil War and crisis. We're going to predict that in certain parts of Germany, I think they took like 1 million refugees. Certain parts of Germany, that in the next generations, we're going to see more Middle Eastern DNA. And we can explain those changes in gene or allele frequencies among modern Germans due to a migration from a different region, right? So this is where you have populations migrate, and you have interbreeding neighbors after a new population.